And next up, I'd like to bring up Natalie Graham, who will be talking about introduced arthropods. Just make sure there's a concert going on outside.
I'll just project.
You did.
The button. Hello, everyone.
Hello.
So we're going to go Malca now and look at some terrestrial biology. And I am an assistant professor in the biology department here at UH Hilo. And I'm a molecular ecologist, which means that I use a genetic lens to understand species biodiversity and interactions. So we are living in a very changing world, and part of the reason why I use molecular tools is to be able to study biodiversity quickly in response to these changes. And some of those changes feel even more pertinent here on Hawaii as an island, because we have a lot of endangered species, we have a lot of extinctions, we have a lot of non-native taxa that are moving in, and we have some biotic homogenization. And so we want to be able to measure that as fast as it is. We want to be able to measure that and get in front of it. And so there's some advantages to this type of molecular biodiversity assessment, but I want to emphasize that this is the type of method that you want to do in partnership with traditional monitoring using taxonomy. One thing that we can do is we can use this for biosecurity. So we can have this rapid response, perhaps in response to a new introduction of species, even before they become established. So we can detect them using something like environmental DNA. We can also look at species that look morphologically similar, but in fact they're cryptic species, and we can tell them apart by their genetic signature. As well, we can look at population level differences, so using the genetic signature to understand how there might be variation across geography or other features that might be separating populations. We also can look at juvenile taxa and distinguish what species they are, even before they can be taxonomically identified. And this is especially important with arthropods. For example, caterpillars are quite difficult to tell apart whenever they are in caterpillar form, compared to when they're in their moth or butterfly form. And there are a lot of species that are undescribed. So sometimes people think that we have all of the species described here in Hawaii, and that is very far from the truth. This is also an emerging field of non-destructive biodiversity assessment, so we can use environmental samples from different substrates such as soil or water in order to get at the DNA signature and figure out what species are present without ever having to take a sample from the specimen or kill the specimen itself. So I'm going to tell you a little bit about some work that's ongoing in my lab and also was the emphasis for my postdoctoral fellowship. And so we call this the early detection of invasive arthropods. We're trying to develop a system that could be deployed. This is in conjunction with the Department of Defense. And the idea is to partner with the Oahu Army Natural Resources Program as a means to answer questions for them, to help them with their monitoring. So our first task was to think about what type of methods we can use as an efficient sampling method in order to capture really the entire arthropod community, and also to think about if we can bring in novel methods such as environmental DNA to improve that type of biodiversity and biosecurity. We're also interested in trying to do this very rapidly and also do it just using a genetic signature. So we've built a classifier to help us to classify things as native and non-native just from the DNA. And finally, we're interested in understanding what properties of the community make them more susceptible to invasion. So this is actually across multiple archipelagos in Guam and the Marianas, Okinawa, and in Hawaii. And today I'm just going to talk to you about our preliminary results, which are from the island of Oahu. And just I'm going to share the results from task 3 and 4. So for this study on Oahu, we set up 3 different elevation transects. And each elevation transect is a different part of the range. So we have one at North Range, this is KTA. We have one at the East Range on Schofield, and South Range. This South Range is Pu'uhapapa. And importantly, these transects represent a gradient from a fairly intact forest in the high elevation to very degraded right on the edge of where the military operations are happening. We collected arthropods using multiple different types of methods, and some of these are just pilot studies to determine if we can bring in different types of environmental DNA. So we are sampling environmental DNA from flowers to look at pollination interactions. We're sampling spider gut and spider web interactions. The web itself should act as a natural aerial sampler to be able to get at the arthropod community without having to take any type of destructive sample, or without even having to put up an aerial sampler as a trap for the aerial DNA. We're also looking at leaf litter. We're freeze drying leaves, and we're doing leaf washes to look at the biotic associations for particular plant species. And we're doing traditional flower visitors using yellow pan traps, eDNA sampling from streams, understory vegetation associates, just vegetation beating, and malaise traps. And then also eDNA from Drosophila baits. Part of the Oahu Army Natural Resources Program is tasked with monitoring rare and endangered species such as the picture-wing Drosophila. And so we want to come up with methods that help with their monitoring program. Today I'm just going to share results with you for the traditional collection of arthropods and the DNA metabarcoding that we did. I'm not sharing any environmental DNA results today. So there's been some lessons that have been learned along the way for how to create accurate biodiversity assessment using molecular methods. And some of this were lessons that I learned through trial and error in my PhD, just because this is kind of a complex and emerging field. One of those is that it's really important to have a barcode reference library. A barcode reference library is a DNA signature from a voucher specimen that you have an identification for taxonomically. And then you voucher that voucher specimen DNA as well. And then when you go out and collect the DNA and you only have the DNA signature from a stream, you can match it to the DNA signature of the specimen in a museum or in a personal collection. We also have found that it's useful to use multiple different molecular markers. So these are regions of the DNA that can amplify at different success rates depending on what the taxon is. So a beetle may amplify better than a bee, for example, for that particular molecular marker. And so it's important to have a variety of these markers. A lot of what we use are mitochondrial markers because they're evolving at a rate that is useful for delineating species. We also employ technical replicates and PCR replicates so that we can be assured that the result that we're getting back is accurate. And we bioinformatically remove things like pseudogenes. So some regions of DNA, the mitochondria, are copied into the nuclear region. This is just a process of evolution and gene evolution. Once those have been copied into the nuclear DNA, they don't actually have any function. And so they tend to evolve on their own, and they can evolve to be quite distinct and different from the mitochondrial DNA that you think that you're looking at. And so it's important to remove those pseudogenes so that you don't compare apples to oranges, so to say, when you're trying to determine what species that you have. Otherwise, you'll have very high inflated estimates of biodiversity. Also, using species delimitation. So there's multiple different types of algorithms that help us to cluster what are these molecular species into putative species, either based on distance between the species or based on some measure of their relatedness. So looking at like most recent common ancestors. And then also we would really, really like to have abundances. You really can't do community ecology without having abundances. It helps us to understand the overall diversity, it helps us to understand evenness in the community. And so in order to get abundances, well, we need to be careful that we're not running into this PCR bias problem. We need to make sure that we have removed our nums. And so what we found is that we can actually get pretty good estimates of relative abundance from the sequence reads, but we have to make sure that we don't introduce any bias, because as you can see from this graph, there are some differences between how certain taxa amplify. Okay, so now that we have our DNA, we'd like to be able to incorporate that with environmental informatics. We want to be able to tell something about the community. And so either we have this sample from our traditional sampling or from eDNA. We're going to run it through a barcode reference library to figure out what species that it is. We'd like to include some information about how those species are interacting. So we're going to mine the literature and figure out, is this an introduced species? Is this not? What type of biotic associations does it have? Might use our machine learning algorithm in order to predict what that introduction status is as well. And include any kind of environmental covariates for what type of habitat it is or what the climate is like. All of this is aimed at trying to get some sort of dashboard or some sort of deliverable that's useful so that we can predict what the introduction status is going to be for that organism. If it's already arrived on our island, how much is it going to spread, or if it's going to be a pest on another island soon. And a lot of this work has been going on with Sierra Martinez and at Berkeley, the Data Science and Environment Group. There's also some applications for AI that we've been looking at and exploring for biodiversity assessment. One of those is looking at these photo vouchers that we've been taking. So we do non-destructive DNA extraction of our whole organism sample, or whole-bodied organism sample. So this is a typical sample that you might collect from a vegetation beating sample. And I can look at this, and as an expert, I can tell you that that is a Pageopalus spider, because I recognize what it looks like morphologically. Can I train a neural network to recognize that that's a Pageopalus spider? Perhaps. So far, we haven't had too much success with being able to get the clarity of images for this, but we have used it to help us count mites, which we can differentiate the mite from other debris in the soil sample and count the mites rather than counting them by hand. Another AI detection method is using machine learning in order to separate out species based as native and non-native. And if you look at this lovely golden clade right here at the top, that is our spiders here across the Hawaiian archipelago. There's quite a large radiation of spiders that are endemic, and they are each other's closest relatives. They have evolved here, and there's quite a short distance between them because they're each other's sister species. If you compare that to the introduced clade, they have a very big genetic distance between them because they evolved somewhere else and then they colonized here. And so they're not each other's closest relatives. And so we can use both genetic diversity as well as genetic distance to separate taxa putatively into these categories of native and non-native if we have some number of them that we can assign ourselves as training data. Okay, so a little bit about the results from those transects. The first thing that I want to show you is that we have some differences between the OTUs. And what's interesting here is that this range edge site is actually a disturbed habitat, but the alpha diversity as measured by the number of these molecular species is higher than our more intact habitats. And why is that? That's really one of the primary questions that we're going to look at. We can also look at the overlap of the number of OTUs that are there. So this is a Venn diagram that gives you an idea about what the beta diversity is and what species are shared between these different habitat classifications. Now, some of the habitat difference, actually the majority of habitat difference that you see here, is the difference between the Ko'olaus and the Waianae's mountains. So the music habitat that we sampled was in the Waianae's, and then the Ko'olaus were the high elevation wet forest. So some of these habitat differences that I've classified really just have to do with geography as well. So who did we detect across all sites? So there was 8 species that we found that were shared across every site, both native and the disturbed habitats. And these are some of the usual suspects. So in invasion biology, we have this amphipod, we have fruit flies, the oriental fruit fly.
Wait, leave it, don't touch it. It's gonna push the table.
The dancing moth. These Crambidae moth, which are our only putatively native species. We have this ant mimic spider, so this is a salticid spider that looks like an ant, introduced. We also have columbella, which are kind of the non-insect and close to insects, and the base of the food web. And then also the spotted-winged Drosophila. One thing I want to point out is that this species was not a high enough taxonomic match to know what it is, so we don't actually know what species this is, and we only have it to family level, and we're only using the machine learning to predict that they're native. The other thing is that that assignment to the ant mimic spider was only at 95%, and that was actually an incorrect taxonomic assignment based on what was in the reference database. We don't have a voucher for it in the reference database, so the assignment could only be at 95%. But going back to look at this, and based on our knowledge of what's at the site, we were able to give a correct assignment. And so we went back in and reassigned this as the correct species. So getting back to that question of thinking about how things vary by elevation along these elevation transects. And we see this pattern where we have the proportion of native taxa that increases significantly with elevation, which is something that we would expect. Our mountains are acting as refugia for native taxa, and the lowland areas have more human disturbance, and so therefore have more of these introduced species. Remember that pattern when we looked at the number of OTUs that were there at that range edge site that was a disturbed site? Well, that is actually what's driving this interesting pattern where we see OTU diversity decreasing significantly with elevation. So really that's primarily because we have this homogenization happening, but also there's still native taxa that are at the range edge, mostly inside the aluhe. The aluhe is acting as a refuge for them at that range edge where all of the military operations are happening. We can also break this down by different sampling method and also by site. So I had them classified as habitat, but we can look at the particular sites that we measured. And just one thing I wanted to point out was that this KR site is actually totally dominated by strawberry guava. Strawberry guava does not support a lot of arthropods, and it also, the arthropods that are there are mostly non-native. Okay, so some key takeaways. One of those is that this is a method that can help us to pretty rapidly determine what taxa are there. It's still a work in progress because we need to work on building up our barcode reference library to make it more accurate. And that's some work that's going on currently in my lab. We also do see this higher richness at this range edge sites, likely because we have this infiltration of these non-natives. And so it's bumping up the higher richness. Machine learning is actually helpful in assigning what the status is of species. And then I kind of snuck another result in here into my key takeaways because I really wanted to understand, are these sites different between the transects, or is it that the sites are different between low and high elevation overall? And so I ran a permanova, which is basically like an ANOVA with Bray-Curtis distance as the response variable, because I wanted to include habitat and site and elevation as these predictors. And so what this model allowed me to do is to understand really that mountains are acting as refugia here. So that's actually what's protecting our native biodiversity, is these higher elevation areas. And within the system that we went to, a lot of those higher elevation areas are actually protected areas that are on Army land. And so in a way, we're protecting the native forest by having just less people moving through it and having the land be reserved for just intact forest. Okay. I just want to also point out that these efforts are really meant to be driven by what deliverables are needed by the community. So we have this in multiple different implications for genetics research here. And also, arthropods are super cool. And actually the Powell-Alkalani doctrine recognizes the role of arthropods as native species. And so we want to develop things along with our partners that are useful tools and also can answer questions that are of basic science. Would like to be able to do both useful applied science and basic science as well. And we recently wrote a paper about this, thinking about how we can do indigenous data science so that the biodiversity stays here and that we keep things within a data pipeline and practice the care and fair principles. And with that, I will take questions. Dr. Knoop.
Great talk. This is more like a speculative question, but if I could ask you to project forward 50, 100, 200 years from now, do you think that this great diversity of arthropods that are introduced, that you find so many of the lowland and disturbed areas, but less so in native high elevation intact, are they just going to move up slope, or is there some kind of invasion resistance that we think will kind of hold them at bay in terms of their ecology, aside from human management?
That's a great question, and one that I wrestle over with, typically with a beer at So really the question was about, is there a biotic resistance hypothesis for communities that can kind of keep some taxa out? Are there some sort of aspects of communities that allow them to resist change? And my understanding from kind of what we see currently with the arthropod community is that there are certain species that seem to be able to infiltrate, or even certain groups that seem to be able to infiltrate, and then others that kind of stay on the edge. And I think that probably has to do with sort of an island effect, and the fact that we have a very depauperate arthropod community in some areas. So there's some groups that have radiated here, and there's just explosive number of taxa and very, very large speciation events that have happened. So once they got here, they kind of filled every single ecological role that you can think of. The spiders are a good example, and so they're so ecologically dominant and already doing kind of doing life multiple ways of the way that you can fill those ecological roles, that there's not a lot of room for some of these invasive taxa to come in, so they're basically outcompeted and they don't establish. Would increased topical pressure eventually lead to, or will there be other, indirect competition that leads to the loss of species, and then opportunistic species can come in, even if there already is biotic resistance for some taxa? Yeah, I mean, that's part of the problem. And you also have catastrophic events, so things like even hurricane, or looking at rapid ohia death. You have these things that are triggering things where we don't actually know the stochasticity of how the community might change after those things. Yeah.
I'm just curious, so you said that with the PCR, some species underwent PCR faster and more prevalently than others. How did you keep that in mind when you were measuring relative abundance?
That's a great question. So the gold standard is to actually make what we call a mock community. So you sample individuals and then you know what the known starting tissue is. And that graph that I showed was from a mock community. So we actually would then know that the amplification of the starting tissue for this particular group, and sometimes it's that there's a slope related to a particular family or a particular genus, and it always goes up kind of within that group. Sometimes it's a particular insect order. They always have kind of an increase in the number of reads depending on how much starting tissue you put in. But the idea is that you put allotted amounts, and then you know what the PCR response is going to be. And then you can use those slopes from your mock community, so you can use how many sequences you get from starting tissue to adjust for what the relative abundance is. The shortcut way is to, which is what I did here, is to convert things by doing a transformation of your data. so that you eliminate some of the, so you can rarefy your data based on the affordances, and then also do a transformation of the data that eliminates some of that. And that's been written up as well. Yeah, no.
Natalie, so, right time.
Yeah,
Let's get on.
sorry.
That's okay.
Yeah, will you still be here after?
I will be here, yes, and for the party afterwards as well.